Yep. Okay. Hello, everyone, and thank you for joining for the webinar today. So presenting on feeding our gut microbes to prevent chronic disease. And I'll also talk a little bit about personalised nutrition as well as part of the presentation. So I'll first initially start with a little bit of background about the microbes, what their functions are, and health effects. I'll then talk about feeding our microbes and the impact of different dietary patterns, particularly fat, protein, and carbohydrate. And then talk a bit about the opportunities for dietary fibre and whole grain cereals in improving our gut and metabolic health. So our gut microbes are a collection of bacteria, archaea, and eukarya that colonise the gastrointestinal tract. We have tens of trillions of bacteria in our gut, and a majority of these are located in the large intestine. And a majority of the microbes are generally beneficial or benign. So as the technology has advanced, particularly in the last 10 years, and it's become a lot cheaper and I suppose more comprehensive in how we can quantify our microbes. There's been a huge amount of research that's been done and across the impacts of microbes and basically all health-related endpoints. A lot of the work is focused on health in relation to the digestive tract itself, such as colon cancer, inflammatory bowel disease, but we're growing our understanding of how the microbes influence the brain in terms of the gut-brain axis, influence immune response, such as respiratory disease, also a link with the gut and cardiovascular disease, peripheral vascular disease, and also bone health and arthritis. But substrate supply is a key factor in shaping our gut microbiota and the metabolites that are produced. So there have been a lot of associations developed linking the gut to various things such as geography. When someone moves from one country to another, the microbe composition changes accordingly. As we get older, the diversity of the microbes decrease. Drugs such as antibiotics have an effect on minimizing the amount of gut microbes and how they function. And exercise and also disease also have impact on the gut microbiota. But the diet is a key factor, and in all those examples, diet plays a key role in why the gut microbes change in the different ways due to those environmental factors. So just to basically run through how the dietary, particularly the macronutrients, influence the microbes in our large bowel. So the carbohydrate, protein and fat are primarily digested in the small intestine, but small amounts of these nutrients pass through into the large bowel where they can undergo fermentation. So carbohydrate that passes into the large bowel is what's classified as dietary fibre, but protein and fat also become a fuel for the microbes. So once these nutrients are in the large bowel, they can have beneficial effects or harmful effects in terms of the microbes that break down, particularly dietary fibre, produce short-chain fatty acids such as butyrate, propionate, and acetate, which have beneficial effects. Whereas there are a whole range of harmful metabolites that can be produced, such as lipopolysaccharides, secondary bile acids, and breakdown products of proteins, such as phenols, cresols, and ammonia. But the gut also has a barrier that helps defend the microbes and their products from passing into our body. This includes a cellular barrier, so the colon cells themselves are joined by tight junctions that prevent microbes from passing through. They also produce mucus, which is another barrier, and there's recent evidence showing there are also defense peptides produced by the colonocytes that pass into the mucus barrier, which can specifically break down the microbes that are present there. If we look at the main nutrients, so a high-fat diet, what does this do in the large bowel? So the higher the fat intake leads to higher amounts of fat that reach the large bowel and as a fuel for the microbes. And as a result, this increases the amount of pro-inflammatory markers. It stimulates the production of lipopolysaccharides, which are absorbed into the blood and induce inflammation systemically throughout the body. We see changes in microbes, such as a reduction in Bacteroidetes, increase in Firmicutes, and also opportunistic pathogens. And many of these changes are linked towards the development of insulin resistance. On the other hand, high protein, protein drive the increase in bile acids, phenols, and cresols, and a reduction in the carbohydrate fermenting bacteria. They've also been shown to stimulate TMA, TMAO, particularly high intakes of red meat. And this metabolite is linked with increased heart disease risk, and with the other protein metabolites linked to increased colon cancer risk. Dietary fibre, on the other hand, in the large bowel, some dietary fibres stimulate the production of short-chain fatty acids, acetate, propionate, butyrate. As these are acid molecules, they lower the pH, which is creates an unfavourable environment for pathogenic bacteria. And they also reduce the prevalence of bacteria that otherwise produce the toxic protein metabolites. Dietary fibre also improves gut barrier integrity and promotes mucus production, and it also has systemic effects in reducing metabolites such as the TMA, TMAO, and also there are a wide range of short-chain fatty acid systemic mechanisms, which I'll explain in the next slide. So here, this is quite a nice summary of many of the different effects of the short-chain fatty acids produced in the large bowel that can either have direct effects on the colonocytes themselves and is linked with a suppression of tumorigenesis, particularly in the colon, and that's probably one of the longest, most researched pathways and mechanisms. And it also does this through reducing inflammation. But in the short-chain fatty acids also stimulate the colon cells to produce gut hormones such as PYY and GLP-1, which then go into the circulation and can influence energy regulation, or the short-chain fatty acids can directly influence the brain, the liver, and pancreas as well in other mechanisms of energy regulation. And the short-chain fatty acids also influence inflammation in adipose tissue, particularly in people with obesity. And more recent studies are showing a link between elevated short-chain fatty acids in the blood and reduced inflammation in lung and respiratory conditions such as asthma. But dietary fibres are not all the same. And particularly as we consume a diet that is made up of many different types of foods, we get a whole range of different types of fibres into our large bowel. And here I've characterised them into three main groups. We have the viscous soluble dietary fibres such as beta-glucan, pectins, and psyllium, which when they absorb water, they form a gel. And this can has been shown to bind cholesterol and be part of the mechanism for lowering cholesterol levels when these fibres are consumed. but they can also slow the gastric emptying. So it means the food stays in the stomach longer. This is linked with lowering of glycemic response, and also in making people feel full for longer can contribute to reducing the amount of food or energy intake that they have. The soluble dietary fibres, on the other hand, they don't have that strong gelling capacity, but instead they help with regularity, such as fructans and arabinoxylans that are commonly found in many of the cereal type products. And then we have insoluble dietary fibre, such as resistant starches and cellulose and hemicellulose, which also contribute to bulking. But the other important thing to be aware of is that not all of these fibres are well fermented by the microbes in our large bowel. And some of these are fermented better than others. And these include the beta-glucan, the fructans, arabinoxylan, and resistant starches, whereas fibres such as cellulose and hemicellulose, commonly found in wheat, are very poorly fermented. So to stimulate the short-chain fatty acids, it's important to consider those fibres that are well fermented. And for the next few slides, I'll be mainly focusing on the work that we've done in terms of fructans and resistant starches. So for fructans, these are, out of all the dietary fibres, the most heavily studied, and there's quite a solid body of animal and clinical data showing the health benefits of fructans. So in relation to bowel health, they're showing that they're well fermented, produce high levels of short-chain fatty acids, improve laxation, and linked to reducing colorectal carcinogenesis. They also have metabolic health effects. They lower the glycemic impact of foods, and when consumed over a longer period of time, they reduce or improve insulin sensitivity and also reduce body weight. And they've also been shown to have bowel health effects of improving mineral absorption. But most of this work's all been done with inulin and oligofructose, which are derived from chicory or Jerusalem artichoke. So other fructans from other plants are less clear. And so we conducted a trial, it's an animal feeding trial to evaluate the gut health and metabolic effects of cereal fructans, particularly barley grain and wheat stem. And because the structure of these fructans is quite different to that of oligofructose, so that's why we hypothesise that these shorter DP lengths of the fructans would have potentially greater functional effects. The first finding that we had was that generally all these fructans produce the same amount of total short-chain fatty acids in the digester, and they had a similar lowering of the pH. But it was interesting that the cereal fructans increased the propionate levels, specifically higher than what oligofructose did. And these animals also found a lowering of their visceral fat weight, which was not seen with the oligofructose. So something specific to do with the structure of the cereal fructans. We also found that the wheat stem fructan in particular lowered GIP, which is a hormone involved with glucose regulation, and this was significantly different from the other groups. And we also found that although all fructan diets improve mineral absorption and retention in these animals, it was the wheat stem again that showed a superior effect in promoting calcium and magnesium retention as well. And so this really highlights that not only is different fibres have different functional effects, but even within a subclass such as fructans, you can get more favourable effects, particularly if certain short-chain fatty acids are stimulated. But it's also important to put this in the context of, I suppose, individuals and the population level, that not everyone can tolerate fructans, for example. And here I've put up the statistic that 1 in 7 people are affected by irritable bowel syndrome, and a large proportion of these people have alleviation of symptoms when they avoid FODMAPs in their diet, and fructans are one of those key components. So again, diets, and particularly a fibre and a fermentable type of fibre in this instance, that has potentially a wide range of very favourable metabolic effects for a reasonable proportion of the population, they can't get that benefit. So it's important to be aware of that. And the final point there is that caution should be taken when recommending a low FODMAP diet, because particularly for people with irritable bowel syndrome, if they're excluding a large number of different, particularly cereal foods that contain these fibres, the diversity of fibres that they consume is low, and as a result, they get a reduction in number of microbes, and particularly beneficial microbes that aid fermentation. So the next few slides I'll shift towards the story with resistant starch. So resistant starch is the starch that escapes digestion in the small intestine and passes into the large bowel, where it's fermented. And we found that it, in particular, stimulates the production of butyrate. This study that I've included here is one where we fed animals a Western diet high in red meat and fat for 4 weeks. And we were particularly interested in whether the impact of this Western diet on increasing DNA damage in the colon of these animals, which is linked with increased cancer risk. And we use this COMET assay, which shows the image where if there's a perfect circle, the DNA of these cells has very little DNA damage, whereas a longer comet tail shows considerable damage. So if we look at on the left, the 0% RS diet is just the animals that were fed the Western diet. They showed quite high levels of DNA damage to the colonocytes lining the colon. But as we increase the amount of resistant starch in the diet, we see a stepwise reduction in the amount of DNA damage. And this was correlated nicely with the amount of butyrate that was in the digester of these animals. And this next study is one that was conducted in pigs, and this highlights the potential synergistic effect of different fermentable dietary fibres. Important to note because we don't consume generally one type of fibre alone. So in this case, the animals were fed either a low or high fructan diet or low and high resistant starch. Both of those fibres in their own right stimulated the bifidobacteria, but when the fibres were consumed together, we got an even greater effect on stimulating this beneficial microbe. This other study has only just recently been published and is quite an interesting one as well, that it highlights that resistant starches in the category of different types of resistant starches can have different effects on stimulating fermentation. So they used 4 different types of starches. The one on the left is just digestible cornstarch, so very low in resistant starch. And then 3 different types of resistant starch. And if we look at the potato resistant starch on the far right, we see that this type of resistant starch has no impact on fermentation. So the microbes aren't able to break down this resistant starch at all. Whereas the 2, the maize resistant starch and the tapioca resistant starch have actually different effects. With the maize resistant starch, increasing the numbers of E. rectali bacteria, which produce butyrate and increase the butyrate levels. Whereas a different effect was seen with the tapioca-resistant starch, where we see a specific increase in the P. distonus, which stimulate the propionate levels. So this is quite a beautiful, elegant way to show that even within the different types of resistant starch, you can get very targeted effects on microbes on the short chains that are produced, and potentially metabolic changes as a consequence of that. And then this is a figure that's come from work from our group, where it was an intervention in 46 healthy individuals that consumed NSP, so non-starch polysaccharide, but resistant starch was increased to 22 grams per day, and this was done just through whole foods approach. But the thing I wanted to point out here is the variation in individuals in this group. So they're ordered on the x-axis in order of their increasing butyrate concentration in their feces. And it's quite striking to see that just across the whole cohort of volunteers in this study, they have a background butyrate level that ranges from very low of 5 millimoles per kilo body weight right up to 30 millimoles per kilo body weight. It's also interesting to see if we look at the far left again, that small cluster at the bottom, by consuming 22 grams of RS a day, they actually didn't respond that much to that level of RS, with generally small increases in their butyrate concentration. And even for some individuals, they get a decrease. And then at the other end of the spectrum, at the high end, we see most individuals get a decrease, even though they're consuming high levels of RS. And that may be explained by the fact that you've got individuals in a dietary intervention trial, they're being contained in what types of food they ate. And if before going into the study, they were eating much higher levels of dietary fibre, then just the controlled nature of the study, that they're actually consuming less fibre than what they would normally be doing. But it is interesting to note that for a majority, so in that middle band, most of the individuals showed a considerable increase in their response to producing butyrate. And overall, the average for all individuals was a significant increase in the levels of butyrate. And this is another very recent study that shows a very interesting effect, where depending on whether a person has this particular balance of microbial species, so if they've got a low Prevotella to Bacteroidetes ratio, which is this ratio is quite common in people consuming a Western diet, that going on a high fibre weight loss diet, they generally, in the blue line, didn't show that much of a decrease in body weight. However, those that have a high Prevotella and Bacteroidetes ratio, they respond very well to a high fibre intervention and lose the most weight. So an interesting effect of the background bacterial composition can influence how responsive they are to an intervention. And in this case, it was looking at weight loss. So a major challenge that we have is that people still aren't consuming even the recommended levels of dietary fibre, which is 25 grams a day for females and 30 grams for the males. And for Australia, where I'm from, about 80% of the people are not consuming the recommended levels of dietary fibre. So we need to come up with new approaches to try and address how we can increase people's dietary fibre, and particularly fermentable dietary fibre intakes. And the other anomaly in Australia is that the type of dietary fibre that we can commonly consume is predominantly from wheat and wheat bran, which doesn't contain a lot of fermentable fibre. So need other strategies of ways we can improve, particularly wheat and its fibre composition. And in terms of resistant starch, the levels are very low in cereals. So rice, oats, conventional wheat and barley, resistant starch levels are very low. And that's where we've developed the homolyzed wheat variety that's high in resistant starch to address this need, with the resistant starch level being high in the refined and whole grain varieties. And Xiongyi Li will talk about this in his presentation today. We did a clinical trial and showed that the wheat, bread made from wheat, high in resistant starch, dampened the glycemic response compared to conventional breads, whether they be wholemeal or refined. And this creates a great opportunity for increasing the amount of resistant starch in staple foods such as bread and noodles. And we've recently completed a study looking at the gut health effects of homolyzed wheat, and I'll be looking forward to presenting that very soon. So just to conclude, substrate supply is a primary factor influencing the metabolites of fermentation in our large bowel. And diets high in fibre, such as, in particular, resistant starch and fructans, can improve health and obtain other metabolic benefits. The digestive short-chain fatty acids are affected by the type, amount, and combination of different dietary fibres, and this can also vary between individuals too. And finally, whole grain varieties that contain elevated levels of specific dietary fibres, such as resistant starch, provide us with an opportunity to improve the gut and metabolic health of people at the population level. And with that, I'll just like to thank all those who were involved with the work that I've presented today, and thank you for your time, and happy to take any questions.